back. Just for part B, but the part A write-up is still due whenever it was due. Right? Okay, so cellular respiration. Now we're not talking about cell respiration in general, just the aerobic part, because we already did the anaerobic part in a different day. All right? Come on. I may not get far enough to do the animation for cellular respiration that I did for photosynthesis, but it's in the Bioflix folder. Look at animations in biology and look at Bioflix. They're really good. All right? Yes. What? Well, okay, about that. Investigation 10 Part A, it said there was a design due this weekend. All right? I'll make it due next weekend to be nice because I never actually talked about it. But basically, you have to write a design, which is basically just writing what it says in investigative labs, to be honest, to actually quantify the primary productivity of the plants. So every week, well, we didn't do this week yet, but we've been killing plants every week, right? And so every week we kill the plants and we measure the biomass. So we measure after 1 week, we measure after 2 weeks, and we're going to measure after 3 weeks. Are you with me? So we kill some of the plants. So what you want to do, right, is write a design that uses the biomass, the dry biomass of the plants, are you with me there, to quantify the primary productivity somehow. That's how to do that is actually in the investigative labs write-up, all right? And that's what you guys have to do. Hi, no, we're in the middle of something, so if you need to talk to them. Or? No, actually, I need to speak to you. Okay. All right, so I'll be right back. Huh? I'll wait for you. Well, it's 20 minutes. Okay, then I won't wait for you. Yeah, it's just going to be... So, sorry. You want me to come by? It's just 2 seconds. Okay. That's fine. Yeah.
Yeah. But what do you do? I can't.
So I don't think we'll even have time unless you want to do it in 15 minutes, change the design, bring it to tomorrow. You guys have to, pero I do it. I can do it. Something that won't take long.
You need to cut the potatoes side. You guys want to drink my coke? No, it's like small. Or just like the floor.
Do you think it's temperature or try to take long? the temperature in some period 30 minutes before we get here to 30. So
All you can do is
all we have to do is like flip it. Yeah, flip it. Should we do that?
We should do everything today.
Okay, let's do it, but I don't want to die too. But let's make
no,
this
but
steam.
the
Can you do it in fifth period though? Or you're doing homework in fifth period?
No, I do everything.
The thing is, I need to study, but I don't study at all.
That's okay. Then I'm coming to let's just get it out of it today.
at the same time, two of us working on
Exactly, just.
No, I'll write it on.
Okay, that's perfect. Hey guys, let's go. So, we already talked about this thing right here, which is glycolysis, this little monster. Alright? But, okay. It's Mr. Jimenez, everybody. So, but that's anaerobic. Probably preceding, right? That's that. Thank you, Mr. Jimenez. I'll take care of it today, okay? Alright? And, so this thing right here is what we have to talk about, okay? The other parts. So notice that at the end of anaerobic, you get this pyruvate, right? Now, this is a bit of a monster here, and I don't want you guys necessarily to remember, understanding the actual steps goes a little bit beyond the scope of what we need to do in Introduction to Biology class. This is biochemistry. But I want you to see the big picture of what's happening. So, the pyruvate undergoes a little chemical reaction that, notice, consumes, or actually produces another electron carrier right there, NADH. So, at the end of glycolysis, we had a net 2 NADH production and a net 2 ATP production. That was our net energy at the end of glycolysis. It was miserable. I mean, remember, fermentation was all about recycling these to use that to repower so you can keep doing fermentation, right? Because you're not going to be able to use these unless you do the ETC down the road, right? So fermentation does not actually produce more ATP. It only uses up this thing that would otherwise be useless to keep glycolysis going, right? So I showed you that here. Well, it's down the road here, I think. There it is. So you're not actually producing more ATP on this step, right? By the way, this is the yeast one. We talked about that produce alcohol. We are actually only recycling the thing so that we can keep doing that part, which keeps producing ATP. But only 2 miscible ATP at a time. We can't sustain a cell this way, not a eukaryotic cell anyways. So now the pyruvate, through this little process, is converted to this thing called acetyl-CoA. This is important, right? Now notice that that actually consumes ATP right there. 1 per pyruvate, right? So I called it the grooming stage. There's no official name for it. A lot of people put it together with the Krebs cycle. But what happened during the grooming? So let's look at that. So remember, I did this last time. I'm doing it again. We're going to kind of track the reactants, the products, what's actually happening. That's the big picture that you need. So let's look at reactants. We actually had to use pyruvate. Now, I'm only writing this once, but per glucose, we made 2. So it's actually 2 of those pyruvates, right? So it's 2 pyruvates. And then you actually need the uncharged electron carriers, 2, 1 per pyruvate, right? You also need 2 ATP, all right? Products, what do I get? Instead of pyruvate, which is a 3-carbon, look, 1, 2, 3-carbon molecule, I get acetyl-CoA, which is a 2-carbon molecule, right? So I go down to 2 carbons on acetyl-CoA. And of course, since I'm doing this, CoA is just, by the way, a coenzyme that doesn't get used up. It gets recycled. You can see that in a second. But of course, there's 2 pyruvates, so I made 2 of those. But you see how a 3-carbon molecule went to 2 carbon. What does that mean? There's a carbon missing. So what do you guys think gets produced right here? CO2. Very good. All right, so CO2. So then we also make 2 CO2. 1 per, that's the first time we made CO2. All right? Until now, there was no CO2 production. Glycolysis does not make CO2. That's the CO2 you get over here. 2 of them at least so far, right? You also used up those 2 ATP, So you made 2 ADP. And you used up those 2 uncharged things to make 2 charged ones, 2 charged batteries. Now notice what's interesting here is that, yes, I am getting rid of the ATP that I had over here. So I actually costed the 2 miserable ATP that I did doing glycolysis. I just lost them to get inside the mitochondria. I call the mitochondria tax. And then, but I kind of did a trade though. I did get more NADs, and that's okay, because you're going to see down the road, each NAD is going to make more ATP than you would get otherwise. This is actually an what we call like an investment, right? So you bought the apartment waiting for the payoff, because the NAD in the ETC is going to give you more back than those ATP will give you. So yeah, it costs 2 ATP to make 2 NAD, but NAD, each one can make more ATP. Does that make sense? So we're going to see this down the road. Well, let's talk about our net energy production at this point. What is our net energy production at the end of the grooming? How much ATP do we have net? None. But we also have 4 NADHs. So keep track of that. Now let's talk about the Krebs cycle, right? So don't need to know, you see, notice, you don't need to know the enzymes. You don't need to know the intermediates. None of that stuff, all right? But you do need to understand that there are intermediates and that there are enzymes. The guy that discovered this deactivated one enzyme at a time to see that this intermediate accumulating. Because when he deactivated this enzyme, right, this would start accumulating. So that he knew this was that step. Then he deactivated this enzyme, that started accumulating. He actually spent 25 years researching this biochemical pathway to figure out the sequence of all the intermediates and enzymes. Won a Nobel Prize, got a name after himself, the Krebs Cycle. Also called the citric acid cycle, because at the very beginning, you make citrate. Now, notice how interesting. The 2-acetylcolase combined with oxaloacetate, which is 1, 2, 3, 4 carbons. So if you have 4 carbons coming here and 2 carbons coming here, this makes a 6-carbon molecule, right? But you do all of this, and you go from 6 carbon to 4 carbon again. So what happened? You lost 2 carbons. So there you go. 2 more carbon dioxides, and you can see it here, right, and here being produced per spin. But you're going to spin twice because there's 2-acetylcolase, right? So let's talk about the Kreb cycle, right? The Kreb cycle reactants. You start with those 2-acetylcolase. All right, it's really 1, but you're doing it twice per molecule of glucose. Right? You also need a citrate. The citrate is you need 2 citrates, 1 per acetylcholine. Right? Now notice what's going to happen during the process. Okay, look, NADs here, NADs here, NAD here, 3 of them, but twice because you're doing it twice, right? So you're going to do 6 uncharged things. What about over here? We have a production of ATP, so we have 1 ADP, actually it's G3P is produced, and then ATP is produced from that. But skip this step and go straight to ATP because you're not in biochemistry class. But it's 2 because you're doing it twice, 2 cycles, right? And you also produce another one right here. You produce FAD. You get FAD H. Two times you're going to use this electric carrier, all right? And then what you get is a product. Okay, so. And you're going to produce carbon dioxide, but not from thin air, from the actual citrate. So you're going to yank out carbon dioxide from these molecules. All right? Now let's see. By the way, notice that there's no oxygen required still. No oxygen. We haven't used oxygen yet. Right? So let's do the counts. Instead of acetyl-CoA, at the end, what do I have? I have citrates. I still have the 2 citrates. So citrate came in and came out. So it's a cycle. Does that make sense? All right. The NADs get charged to their charged versions, right? So they get reduced to receive electrons. Remember that. Are you following? Pay attention. This is what you need to know. That the net in. This is what you need to know. Reactants, products, and net after the process. All right. So we had the 2 ADPs becoming 2 ATPs, reduction, receiving electrons, being charged. All right. And then we have the 2 FADs becoming FAD H's, FAD H2. All right? All of that, but we also get 2 carbon dioxides. All right? So the 2 carbons that were here are still accounted for. They just went into the air. Now remember that you're actually doing the whole thing twice, and it was 2 carbon dioxides per cycle, so this is actually 4. Right? So it was 1 carbon dioxide here, another one here, but it's 2 spins, because it's 2 acetylcolase. So you make 4. 4 plus the 2 doing the grooming, that's the 6 you make over here. Does that make sense? We're done producing carbon dioxide. Was carbon dioxide produced from oxygen? No, oxygen's not even in the picture yet. Right? Not even in the picture yet. Net energy. We actually made some miserable ATP, too, after all of this. But we also, let's talk about, we had 4 NADs. We made 6 more. All right? So we need 6 more. So now we have 10 NAD H's. We had 2 FADs. Fat H's. Fat H2. All right? And that's the net energy that I have. Okay? To use up on the next part. Okay. This is where the magic, by the way, you can do this on the videos and tutorials that I posted online. This is very similar to the Krebs cycle, except in reversed. All right? We have an activation phase, we have a regeneration phase, and everything. All right? And we have, instead of a capture phase, we have an emission phase. All right? And so it's very similar, but in reversed. Okay? Now the ETC, super cool. So what happens is, I'm going to have time today, but at the beginning of next time we see each other, that is a lecture day, because the next time we see each other is a lab day, complete lab day. We're going to show you a video of this animation, or I'll also send it on Sally to you guys, so you can watch the one I want you to watch. Right? It's the BioFlix one. It will similarly, like the other one I saw, show this in motion, but I do want to talk about it. Now notice how interesting. It's 3 pumps this time. And like before, it's not really pumping. It's biochemical reduction and oxidation, which eventually carries the hydrogens across. But it's 3 pumps this time. But notice how interesting. The NADs, the batteries, power 3 pumps. Each NAD enters here, and then the electron goes down the chain. Now it's like this. I get it. I become electrified. I pump a hydrogen. The next one gets it, pumps a hydrogen. The next one gets it, pumps a hydrogen. So it's like a pump in a wire. So the membrane of the mitochondria is like a wire. Are you with me? And 3 pumps are in it. The battery dumps it, and it goes like a current. Are you with me? And then they're powered on their current. They're like plugged in, the pumps. And then as they're plugged in, at the end of that, there's an oxygen sitting there saying, give me your electrons. Oxygen gets reduced to become water. That's when it gets produced. Right? So every 2 electrons that go through, 1 water gets produced. Now here's what's interesting. The NAD powers all 3 pumps, but the FAD, it's a little fat, so it works out less. All right? So it actually produces, only powers, that's how I remember. It only powers 2 of the pumps. So let's think about this. We got 6 NADs powering 3 pumps each. Or 10 actually. 10 NADs power each 3 pumps each. That's 30 pumps working because of this, right? 10 NADs, 3 pumps each. And then we got 2 FADs, right? Powers 2 pumps each. So that's 4. Total, 34 pumpings take place. 34 hydrogens go to the other side. Are you with me? And like in photosynthesis, they can't pass unless they go to the power plant, the same power plant. So it's like a homologous structure. Are you with me? It's ATP synthetase. So what happens here is that 34 ATPs get made on this process by chemiosmosis, which is the passing of those, using the proton potential that was built with the pumps. Right? Now remember, all these electrons that are powering these pumps came from glucose. And all those electrons that were in glucose came from water. That photosynthesis made it happen, right? And then where did it go back to? Water. So it's a cycle, right? That's happening there. And water gets produced. And that is the ETC, which actually makes this 34 ATP. So the ETC just basically grabs these and transforms to ATP. But notice that each NAD made 3 ATP. So it was worth spending 1 ATP to make 1 NAD. Because you got more. 3 times as much. That's some good returns from the investment in the apartment, right? So that's how life does it. It's basically a power plant. I have a really awesome animation, but no time to show you this time. I'll show you next time. All right? BioFlix and Animations in Biology on the Videos and My Tutorials folder. We'll show you this in more detail. All right? Hold on. One more thought before we leave up is these CAP processes are all about creating the energy that life forms need to maintain homeostasis. Don't forget that. I'll see you guys next class for Lab 5. Make sure you do the pre-lab. See you guys later. Enjoy Thanksgiving. Be thankful that you have an awesome biochemical power plant inside of you, and don't do anything that will not make your mama proud. Now,
I'll actually be here with Thanks, man.
I give them like credit for errors, like it's all fine.
And knowing me, I'll probably like salivate this.
There's two things. First, a few labs and stuff that he has to turn in. Second, I'm behind already, so his grade is actually better with things. And that, so today I have to catch up, so the issues go away. Because her, she also may have a better grade than she has, just haven't graded it. You know what I'm saying? But, yeah, she emailed it to me.
Last week I've been like coming in, catching up, taking care of things.
He reacts
You know that
kind of like when you put in those two right before
So I
you
don't
mix
forget and I
it.
forget everything.
He's kind of like that enzyme that unless you provide the right temperature
To be
and the
honest,
right
you can't
catalyst and the right amount of
I'm
heat,
super airheaded.
he doesn't react. You know, it's when you lower the temperature on alcoholic fermentation, you don't get as much alcohol. It's the same thing.
But Isaac,
Yeah, I've just explained Isaac using biology.
it's true. That's a good analogy. I like this. Isaac, is like always challenging, but always eager to face the challenge. You know what I'm saying? And I actually like him better here than I've had in other classes before. So I think he's, he says he wants to study. I'm like, but the weakest thing is the FRQ. I have to work out. But it's funny because he came to do corrections and he understood what he was supposed to write after the fact, all right? Hindsight is very funny.
Yeah, because I knew all the.
Hindsight is always funny. That's for everything.
I knew the information, just sometimes learning other questions.
Yes, it's true. Welcome to biology.
Like for the math, it was like.
When you actually learn this in biochem, it's like mind warping. It really is.
It's understandable.
This is when most people just go like, fuck biology, I just give up. It really is.
So many notes on that.
It really is.
The thing is, you're getting down biochem. You actually
I
have
remember
to know it
getting
by
a
enzyme.
note, like you know how you use selection.
So you gotta know each enzyme. You gotta know at what temperature each enzyme goes at. You gotta know
For what?
microscopic.
He said turn it
The
on.
day you do it. You're not doing it right
Oh,
now.
Nemo, remember when you had me the first time? Remember when you had me the first time?
Oh yeah, wait, by the way, when are we doing the thing? Today? This happened today. After school. Awesome. This one,
Justin!
this one, missed the test last quarter. Got an F, and I give her a chance to make it up. Now she's making it up.
No, but it's been because of the mistakes between us both.
That's true.
That's true.
To be fair, every time she comes, I am not here, and every time I'm here, she doesn't come. So hopefully today, Alaska.
Laptop, Nemo, you stay?
Yes. Yes.
For sure.
Yes.
For sure.
To like 4:30.
Okay.
All right.
You have an hour and a half.
Okay. Yes.
It's kind of like one of those alpha particles in the hedron. That they sometimes they show up and sometimes they don't.
Exactly.
It's just
Okay. I don't need you to.
All right. What do you need, girl?
Please,
Yes.
calm your shit, okay?